Hi there, my name is Nicole and I'm excited to welcome you back to the Neuro Squad. Today's focus will be on Dr. Michelle Gray, a renowned neuroscientist best known for creating an artificial human genome inside bacteria to be modeled inside of mice. Alright, let's get to know Dr. Michelle Gray. Dr. Gray seems to be a very private person, as much about her life before her secondary education is not shared with the public. She was raised in a small town in rural Alabama where her love for life formed, but that seems to be all she is willing to share with the public. She received her undergraduate Bachelor of Science in Biology from Alabama State University in Montgomery, where she also applied to multiple National Institute of Health research funding grants, which allowed her to do research at her alma mater along with the University of Wisconsin-Madison over summers. She then went to the Ohio State University to receive her Ph.D. in molecular, cellular, and developmental biology. There she worked with Dr. Christine Beatty as her first graduate research student, focusing on using zebrafish as a model organism for the nervous system, where she received the NINDS F31 Ruth L. Kitchelstein National Research Service Award for her work in research. She used this funding to become an independent researcher while still receiving her PhD, and it allowed her to attend important conferences that helped with her education. Afterward, she went to UCLA to work under Dr. X. William Yang for her postdoctoral research in neurodegenerative disorders. She mainly researched multiple sclerosis and Huntington's disease and the way they progress. She now runs her own lab at the University of Alabama at Birmingham, focusing on using mice models to study many different neurodegenerative diseases. One of her biggest contributions to neuroscience was her development of the conditional bacterial artificial chromosome transgenic mice for specifically Huntington's disease, often turned into the acronym BACHD. These transgenic mice don't have huge changes in weight compared to other mice that have been given Huntington's disease in their genome, making them a more accurate depiction of the progression of Huntington's, allowing them to be better models for pharmaceutical options to help treat this disease. The Neuroscience Squad is currently in Neuroscience 175 at the University of North Carolina at Chapel Hill, and Dr. Gray's research on neurodegenerative disorders directly relates to our unit on glial cells in multiple ways. First, one of our learning objectives was to identify why glia isn't really the focus in our textbook compared to neurons, which is simply because glial cells are just now becoming a focus of research and are therefore not elaborated on as much as neurons are in common textbooks. Dr. Gray's research, while mainly being focused on neurodegenerative disorders, plays hand in hand with glia, as neurodegenerative disorders often attack glial cells. Secondly, her research in MS relates to the learning objective from our glial cell unit that asks us to describe the function of the main types of glial cells. We learned that myelin and myelination is crucial for the propagation of action potentials, as they are able to use those myelin channels to flow faster down the neurons. The effect of losing that myelination can make action potentials propagate much slowly, which can have many detrimental effects on the individual, as seen with multiple sclerosis and the way that progresses over time in people. I hope you all enjoyed getting to know Dr. Michelle Gray, a Black female neuroscientist credited with the creation of BACHD and creating her own lab at the University of Alabama at Birmingham.